Dear students, today we will discuss about our second class coming under plant tissue culture, that is somatic embryogenesis. And first we will discuss about what is this somatic embryogenesis. So somatic embryogenesis is an artificial process in which a plant or embryo is derived from a single somatic cell. So it is like that of the here also we are utilizing the capacity of the plant cells, that is totipotency of the cell. Somatic embryos are formed from plant cells that are not normally involved in the development of embryos, that is ordinary plant tissue we can use for the development of embryos. So what are the related technical terms which are using in somatic embryogenesis? That is, somatic or non-zygotic embryo is called embryoid. The meaning of embryoid is embryo-like, okay, and it is an asexual embryo derived from a somatic cell. That is, the embryo developed from zygote is called a zygotic embryo or seed embryo, and the one developed from pollen is called pollen embryo or androgenic embryo. Okay. So before explaining about the somatic embryogenesis, first we will just review what is the normal embryogenesis. Normal embryogenesis we can see which is occurring in the flower. That is, if bisexual flower or unisexual flower, we can see the fusion of male and female gamete during the sexual reproduction occurring in the plants. And from that we can get after the embryogenesis, we can see this developing embryo in the or the dormant embryo inside the seed. So in case of dicot plants, we can expect two cotyledons, and in between the cotyledons we can see the embryo is situated. So in case of pulses, we can see some sort of for example green gram, black gram, all this groundnut. Inside that we can see the small embryo which is in dried condition. So it is showing some sort of dormancy. So then naturally occurring embryogenesis is there. Then why we should do this somatic embryogenesis? So in case in some plants, due to some incompatibility or pollen incompatibility, sometimes the normal sexual reproduction does not occur. So in one more case is there, that is if we are propagating any plant by using vegetative reproductive mechanisms continuously, then we can see some deformities are occurring in sexuality or sexual reproduction in plants. Okay. So in many such occasions we can use for any experimental purpose, we are in need of seedlings or seeds or embryos. What we will do? Okay. And in such occasion also we can utilize this plant tissue culture methods. Okay. And then coming to our present topic. So this is this somatic embryo is actually a non-zygotic embryo. So we can use the term embryoid. So it is an asexual embryo derived from the somatic cells. Okay, clear? Somatic cell embryogenesis or simply somatic embryogenesis is an in vitro formation of bipolar somatic or non-zygotic embryos from somatic explants. That is, by using this somatic embryogenesis, we are producing embryos which are bipolar in nature. That is, it is with a here in this figure we can see this last figure. That is, we can see a cotyledonary embryo is there, and this embryo is with the two poles or two axes. One axis is meant for the development of root system, and the other one is meant for the development of shoot system. So like that of zygotic embryo, here also we can see the embryoid or the somatic embryo is bipolar in nature. So the radicular end is always oriented towards the center of the tissue culture mass. That is, suppose it is developing from callus tissue, that is, the radicular end is immersed in the callus tissue. Okay. And plummular end is always projecting out from the cell mass. So in later slides I will show you the photographs of the same thing. Then the somatic embryos or embryoids are somewhat similar to that of zygotic embryo or seed embryo. The only difference is they originate from somatic cells. Okay. There is no haploid condition because the somatic cells we are utilizing is diploid. So this embryo is developed also similar to that of normal embryo. Okay. Only difference, it is developing from a somatic cell. Okay. Then in vitro somatic embryogenesis, that is we can say, of course, this tissue culture, this is a part of tissue culture. So it is we are keeping these developments or forming these developments inside the lab conditions. That is, first it is demonstrated in 1958 by Renat and Stewart. Remember these names, it may ask for one more question. In 1958, Renat and Stewart, they first performed the somatic embryogenesis. Okay. And on our convenience or by considering the protocol of somatic embryogenesis, there are two types of somatic embryogenesis. There are or two ways of somatic embryogenesis. SE means somatic embryogenesis, that is indirect somatic embryogenesis, where the first the callousing is induced from the explant by rapid cell division, and then from that callus somatic embryogenesis is forming. Okay. Suppose we are using leaf bits, and we are keeping this leaf bits for callus formation in a nutrient agar medium, and from the agar we can develop embryos. So it is indirect. So in case of direct somatic embryogenesis, and which are developed directly from the explant without an intermediate callus phase. Okay. And we can adjust these conditions by maintaining the hormonal level of the media. Okay. So in dicotyledonous plants, the somatic embryos possess or the developing stages are through globular stage, heart-shaped embryo stage, torpedo and cotyledonary stages, as in normal zygotic embryogenesis. So here we can see it's a flow chart, that is somatic embryogenesis, two methods, indirect and direct. Indirect, that is callogenesis, that is callus formation, and then from the callus we can see the embryos are developing. So then callogenesis are followed by embryogenesis. But here in direct method, suppose explant is we are keeping in growth medium and from that explant directly we can see the formation of embryos. Okay. So the only major difference is that somatic embryos do not pass through the desiccation and dormancy phases. That is, normally in case of, for example, green gram, we can see after the formation of seed and embryo formation, the embryo will rest for a period, that is a dormancy period. But in case of this somatic embryogenesis, that is in vitro, especially in vitro somatic embryogenesis process, there is no such dormancy phases. That is, just after the formation of the embryoids, it will grow into a new plantlet. That is a major difference between zygotic embryogenesis and somatic embryogenesis. Then what are the major steps in this somatic embryogenesis? That is formation of embryo, that is induction of embryo, then how the embryo is developing, and how the embryo is maturing. Okay, and after the maturation we can see that it will form into new plantlets. So that is some steps. Then what are the factors affecting in somatic embryogenesis? That is the, of course, this genetic conditions or genotype of the explant. And the growth regulators, especially auxins and cytokinins present in the growth medium. Then nitrogen source. These three are the major factors affecting somatic embryogenesis. And these are some of the examples of explants which are utilizing in somatic embryogenesis. That may be mature leaf, that is very young leaves we can see. Here we can see young leaves. Then hypocotyl of the seed. Then scutellum, inflorescence primordia, here we can see this scutellum area, and it is in case of cereal grains, and all these can be used as explants for the development of somatic embryogenesis. So the somatic cells of explant is of two kinds. First one is called pro-embryogenic determinant cells, that is PDCs. It may ask for one mark question, that is the somatic cells of explant programmed to directly develop to embryos are called pro-embryogenic determinant cells, and this may be utilizing in direct embryogenesis. Then the somatic cells of callus used to develop indirectly to embryoids are called induced embryogenic determinant cells. Okay, and wherever we can see this PDCs and IDCs. PDCs are already predetermined for the embryogenic pathway. They only require the synthesis of an inducer or removal of an inhibitor to resume independent mitotic divisions and to express their embryonic potential. That is normally the somatic cells which is programmed to develop into embryos. That may be in connection with the some tissues related to the embryo formation. It is not embryo only developed from zygote. That is naturally occurring thing, and it is with a period of dormancy, and that dormancy is maintained as a result of presence of some inhibitor substance. For example, seed dormancy we can break by soaking in water. So with the help of the water, it imbibes water and starts to germinate. So this is a simple physiological definition or physiological way we are expressing the growth of the embryo. But at the time, very deeply we are studying about how this germination process is occurring. There we can see this inhibitor substances and removal of this inhibitor substances. When the seed is imbibes water, with the help of the water, the seed removes the inhibitor substance which is masking the growth. Okay, so like that also we can, here also we can use some particular chemicals to or hormones to induce the growth of the embryo. On the other hand, IEDCs require redetermination to the embryogenic state, generally through exposure to specific growth regulators. But PDCs are found in embryonic tissue, including the scutellum of cereals, the nucellus and embryo sac with the ovules of mature plants. So these are already in connection with the zygote or near to or part of the zygote developing area. So it is with some innate ability to develop into. Suppose we are utilizing the scutellum for the as explant. So there is no it may directly develop into embryos, because already that information which is written in DNA is present inside it, because the scutellum is present along with this zygotic structures. Okay, but in other words, that anther suppose we are indirectly doing the somatic embryogenesis, the callus which is we are utilizing is from anther culture, or directly any calluses. Suppose we are utilizing the explantus leaf bit, and from that leaf bit callus is developing, and the callus is not in not the tissue which is present like that of the scutellum or embryo sac or nucellus, because it is very independent tissue. So there we can see that index there, the callus which is in need of some sort of induction. Okay, induction to develop into embryos. So that is the two technical terms, that is PEDCs and IEDCs. Okay, that is determined cells, predetermined, that is pro-embryogenic determined cells. Example, what are they? Scutellum, embryo sac cells, then nucellus tissue, nucellar tissue. But IEDCs, that is induced embryogenic determinate cells, which are present in the normal callus tissue. Clear? And then we will study, it's a 4-mark question, what is the direct somatic embryogenesis and indirect somatic embryogenesis. So just draw the figure on left side and explain it. And of course you should write a small introduction related to this previous slides. Okay. So what is this direct somatic embryogenesis? So in direct somatic embryogenesis, somatic embryos develop directly either from single cells or from small groups of cells of the explant without an intervening callus stage. So there we should use the tissue which are previously just we studied in previous slides. That tissue, suppose it is using for the somatic embryogenesis, it may develop directly. Repeated cell division results in a group of cells which directly develop to an organized structure called globular cells. Here we can see this initiation of somatic embryo development. First we can see this parenchyma, meristem like tissue organization, and it will form a globular or shaped pro-embryo stage. Then it will turn into a globular stage, and then it will it is very fastly absorbing the nutrients from the medium. It will develop or it will do continuous cell division and form a heart shaped embryo, then torpedo, then cotyledonary stage. Okay, and without an intervening callus stage. So further development results first in a heart, once again, that is first in a globular stage, then heart, and then torpedo shaped and cotyledonary stage of somatic embryo. That is direct somatic embryogenesis. So polarity is established early in embryonic development, that is signs of tissue differentiation becomes at the time of globular stage itself we can see a tissue, a polarity is there. Okay, that is two axes we can see. At that last stage, that is cotyledonary stage, we can very well recognize the which one is cotyledons and which one is the root apical meristem. So always this root apical meristem is immersed in the growth media. Okay, then we will study about that is the indirect. Here the that is here some photographs are there. This is actual photographs. That is, this is a globular embryo. Then from that we can see the heart shaped embryo development, and here we can see cotyledonary, of course. Along with the cotyledonary embryo, here and there we can see heart shaped and globular, but on the right side we can see the cotyledonary stage. This is actual photograph of development of direct somatic embryogenesis in vitro conditions. So, what is this indirect somatic embryogenesis? Already we studied, we discussed. Anyway, in indirect somatic embryogenesis, the cells of the explant proliferate or developed into callus in the presence or absence of hormones, growth regulators, that is growth regulators, especially the chemical form of auxins, that is indole acetic acid. So, in some cases the indole acetic acid is more, then it will form the callus formation. Likewise, if it is low, it will produce embryos. So, embryoids develop in large numbers from the callus. The cells and protoplasm producing somatic embryos can be isolated from the medium and cultured again. Just before the embryo formation, we can do subculturing also. That is, we can, callus is very fragile and detach small bits of tissue from the callus and put these callus as explants in different subculturing media. Then, so we can increase the number of embryo formation. Okay, and somatic embryo induction can be done using semi-solid suspension cultures. So, normally it is completely hard medium is not utilizing. In that place we can use semi-solid or suspension cultures. Then sliced out callus pieces can be inoculated into culture, that is subculturing. Okay. Then auxin is to be supplied in 2 steps. Auxin means it is, there we can see this indole acetic acid, chemical forms we can use, or just a pure auxin also, but anyway we should give this auxin in 2 steps. So, in the first step provides a rich supply or more amount of auxin. So, what happens during this, differentiation of localized group of cells called embryogenic clumps will develop. That is, it is along with the callus formation. First the medium is with a more amount of auxins. Okay. And as a result, embryogenic clump formation or the tissue formation occurs. So, in the second step, and for that purpose we can use a different test tube or conical flask with the medium. Okay. And in the second step, these clumps develop to embryos in a medium with a low concentration of auxin. That is, along with the other energy sources and also nitrogen sources. And in the second level of development, we should reduce the auxin content, or we can increase the little bit of cytokinin content also. Okay. And as a result, what happens? Embryogenesis occurs. So, first medium callus formation, and there more amount of auxin, and the second level we will add a little bit of smaller quantity. When we are comparing the previous auxin content, we will reduce the auxin content, and as a result we can see embryo formation. Okay. So, here we can see this plant, the leaf is there and leaf pieces, and we are incubating the leaf pieces, and as a result callus formation. And in this particular medium we can see the more amount of auxin is there. So, we can see from the callus some bits of tissue which is which becomes embryogenic clumps, which turned into just not increasing the callus size, it will turn into embryogenic clumps. Then in the second level we will reduce the auxin content, and as a result embryogenesis occurs. Okay. And likewise we can produce from these small embryos. There is no dormancy period in case of somatic embryogenesis. So, the embryos will directly develop into what? plantlets. And what are the limitations of this somatic embryogenesis? That is, complete conversion into plantlets or poor germination of some embryos sometimes occurs. Then compared to other plant species, active research or somatic embryogenesis involves, that is in case of some endangered species, suppose we are using this method, that research is very low, because according to different characteristics of different plants, the situations or the concentration of the hormone we are utilizing or using may vary. So there is no 100% success in case of embryo formation. Okay. The lack of knowledge controlling somatic embryogenesis, that is the synchronicity of somatic embryo development and low frequency of true type of embryogenic efficiency, are responsible for the reduced commercial application, because it is not always turned into a complete success. But to provide aseptic conditions and other other factors, physical factors, it is economically very high, that value, that financial expenditure is very high. So it won't be 100% success in case of commercial applications. To obtain a complete conversion into plantlets, it is necessary to provide optimal nutritive and environmental conditions. So it is also a hurdle. That are the main limitations. And here we can see some actual photographs. That is, here we can see this, the photograph which is explaining the indirect embryos, somatic embryogenesis. So here in a figure, and it is the close-up view of the same thing, and here we can see that pale color, which is the callus formation, and after the callus formation, normally we will reduce the auxin content and increase the cytokinin content, and as a result, from the embryogenic clumps which is formed in the callus, it will turn into what? Bipolar embryos. Okay, this is the developing this fluorescent photography, that is the D, last one. Okay, and this is the actual photograph, and this is one more example, that is here, this is also indirect embryogenesis. Here we can see the what? Callus, and we are adjusting this growth regulator percentage, and as a result clumps are forming, and these clumps are actually meant for, you can see pale and somewhat dark cream color clumps are there, that is embryonic clumps, and from this we are adjusting the growth regulators percentage. Auxin is reduced and cytokinins increase, and as a result from these clumps, here we can see these clumps, it turned into embryo. And immediately after this, the different types of embryos. Here the last figure we can see the from the that red coloration is there, that is the radicular region. The upper portion is plumulus region, which are develop into shoot system. In the last photograph we can see this radicular region slowly turned to root system. It is a starting stage, and the upper part we can see the leaf formation or shoot, primordia is developing. So this is the indirect, this is also indirect. Okay, and this questions coming under this part is, just you can ask get one more question, that is simply SE, only capital S, capital E. What is this? That is somatic embryogenesis. Just write the definition for it. Then this, what are the different methods we are utilizing in the somatic embryogenesis? That may ask that for 4 mark question. Then you should write about the direct method and indirect method. And one more question, that is for 2 mark question. What is this? Distinguish between PEDCs and IEDCs. Just write the expansion of it and 1 or 2 sentences. Okay. And try to draw and study this particular line diagram and this one. Okay. Study well. If you are having any doubt, please ask questions in Google Classroom. Thank you.